In this video, we will discuss about this HT immunotherapeutic efficacies on tumor mutated neoantigen. In the previous video, we have understood the efficacies of this uh, hepatitis B antigen, hepatitis B virus surface antigen uh, specific mRNA, or hepatitis B virus surface antigen vaccines on this B15 F10 B16 F10 tumor cells. And on that cells we have seen its the efficacies are much better. It is able to suppress the tumors. However, in order to confirm that the results of this HT vaccines is not specific to the single cancer lines, we decided to use a different cancer lines, that is MC38, and along with that they also need to check whether their approach is also applicable to the tumor mutated neoantigens that are expressed on the cancer cells. So for this, in that case, if their approach is applicable to this tumor mutated neoantigen, then they can prevent the final step, that is expressing antigens by inducing this virus, as those antigens are already expressed on the cancer cells. So in order to do check this hypothesis, they used a cancer cell lines, that is MC38, which is known for the multiple tumor mutated neoantigens. So for the control, they use this mRNA-H, and before injecting the tumor cells into the mice model, that is C57BL6, and thereafter they used this VVH to express this hepatitis B antigens on the MC38 cells. Along with that, they also checked it for these tumor mutated neoantigens, and we will discuss what are the different neoantigens that they have tested. And so first, let's just see the results. In terms of the results, they are able to find out that in the case where they use this HT-VAC, they are able to see a maximum tumor suppressions or minimum tumor growth rate compared to the all other cases where they use the antigens, the VVG, or where they only use the virus with the hepatitis B antigens. And these things they have validated by checking the activated immune cells, that is CD4 and CD8 immune cells, that is shown here. They are able to see that the CD4 immune cells are significantly high in the case of this HT vaccine conditions, and at the same time they also see the complete remissions of 33%, which is much higher than all other their control cases. And the immunofluorescence image are shown in this panel D, where we can clearly see the CD4 signals are high compared the CD8 signals are high compared to the all other control cases or all other conditions. And these things they have quantified, that is CD8 cells they have quantified in the panel E, where we can see that it is around 5-fold higher than the all other cases. And the CD4 cells are also high, but those high is comparable to the case where they use the virus only with H surface antigens, and also is comparable to the mRNA-H only where mRNA-H is used. But CD8 results are significantly higher, or comfortably one can say that CD8 T cells are high, and which is the main cells responsible for killing of the killing of the tumors. And thereafter they checked it for the multiple tumor mutated neoantigens, and those peptide sequence are shown in this panel G, which can be seen here. And out of this multiple mutated neoantigens, they checked the efficacies, and they also checked whether they are able to induce endogenous activations of the T cells or not. So for that they checked it using in the splenocytes using ELISPOT, and in all the 10 peptides cases. Here the results for shown for the 10 peptides, whereas the peptide signals sequence are 11. So for the 10 peptide cases, they are able to see that the HT-VAC signals are significantly high, even compared to the case of all the control cases, even compared to the all other control cases. And this is the one of the representative image of this splenocyte of using ELISPOT, and here they have captured the IFN gamma, and these things they have quantified in the panel I, where we can see that in the ELISPOTs of this IFN gamma signals are significantly higher for the HT-VAC compared to all other cases, and results are consistent for the multiple tumor mutated neoantigens, and even the results are comparable where they used hepatitis B virus surface antigens, which is their control case that we have. Those results we have discussed in the previous video. So this in altogether indicates that their HT vaccines can be used for tumor mutated neoantigens also. It is not just specific to one cancer cell lines, or not just specific to the hepatitis B surface antigens. So that's all for this video, and if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.